So today this talk is about a citizen science project called soilSkin, and it's organized into parts. The first half will be about the organisms this citizen science project studies, an introduction on how to distinguish them and their importance. And then the second part will be about how to participate in the citizen science project. So let's begin. First of all, I would like to highlight the importance of soils. Soils are not only the place where we stand, they are a highly diverse ecosystem that is alive. Actually, soils host about a quarter of our planet's biodiversity, which is really a lot. And an important part of this biodiversity are the topsoil communities formed by bryophytes, lichens, and algae that we call biological soil covers. They are important because they cover the soil as a second skin, protecting it and preserving its biodiversity. So what are these biological soil covers? As I said, they are formed by these three groups of organisms that are very different, and they live in the first few centimeters of the soil, the upper part of the soil. And they are also highly diverse. For comparison, we can think about the number of vascular plant species in the Iberian Peninsula, where we have roughly 8,000 species. In the same area, in the Iberian Peninsula, we can find 2,700 species of lichens, about 1,100 species of bryophytes, and an unknown but very high number of terrestrial algae. We really don't know the exact number, but some estimates say that it can be as high as 20,000 number of species, but we really don't know very well these communities, so it's not sure. The number is not sure. It's not certain. So where do we find these biological soil covers? So first of all, we can find them in Mediterranean forests. If you look at this picture here, all these green patches in the soil, these are bryophytes, and these are biological soil covers. They are considered biological soil covers. But also in drylands, if you look at this picture, in here it seems as if it was just a few bushes and bare soil, but all these white patches here, they are lichens. So all these soils are full of life and are very highly diverse communities. But we can find biological soil covers not only in dry ecosystems, also in temperate forests. If you can see here, the soil is completely covered by a green mat that is composed of bryophytes, and in fact in peat bogs, which are common in the north of the earth, and bryophytes are the dominant organisms. And we can even find this type of communities in the Antarctic biome. So the answer is everywhere or almost everywhere, and they even cover 7% more or less of the earth, of the terrestrial ecosystems, the soil. If you think about it, it might seem as a small number, but these are really tiny communities that are barely 1 centimeter high, and yet they cover almost 7% of the earth. I think this is a great achievement for us, such as small organisms. So let's explore a little bit more the main organisms that form the biological soil covers. We will start with lichens. Lichens are made of a symbiosis between three groups of organisms. First fungi, then algae, and also bacteria, so that they are miniature ecosystems. If we cut a lichen, we will see that the most common structure inside the lichen is a multi-layered structure. So here we see this green part. This would be the algae. And then there are two layers of fungi, one below and the other one above, covering and protecting the algae. So this is the typical structure of a fungi or of a lichen. There are many different growth forms of lichens. In the one hand, we have those that are crustose. They are attached to the substrate and it's very difficult to detach them without killing them. They are so attached that they are in the same plane. They do not have 3 dimensions or 2 dimensions. You see them and they are just completely attached to the substrate. They are a crust almost. In fact, the borders of this type of lichen are very diffuse. It is not easy to say when the lichen starts and when the substrate starts. Then we have foliose lichens. These are like leaf-like, and in the borders they usually have lobes that are two-dimensional. So you can see that they are separated from the substrate. They are a different layer on top of the substrate, not completely attached. That's how you distinguish them. These are more diverse in shape than crustose lichens, which are just a crust. And finally, we have fruticose lichens. These ones look a little bit like trees or bushes or maybe corals. Depending on the shapes, they come on a large amount of shapes, very different ones. These are the more diverse, and you will recognize them because they are 3-dimensional. So 3 dimensions, 2 dimensions, just one plane with the substrate for the crustose. These growth forms are very important to understand how lichens behave and how they respond to environment, because they have very different environmental requirements. But they are also important to understand how they influence the ecosystems. And that's why we study these growth forms, because they have key characteristics that can help us understand ecosystem functions of these organisms throughout several different, very different ecosystems. So let's go and have a look at the next group of organisms, which are bryophytes. These ones are true plants, non-vascular plants that produce spores, but they are terrestrial plants, and as such, they are green, bright green. You will recognize them because they have a very characteristic color that reminds us of vascular plants. Okay, so let's go. We have very different types of growth forms also here. Also, same way, very important to understand their responses and their effects in ecosystems. So we have two big groups, liverworts and mosses. Liverworts can be thallus or foliose. The thallus liverworts are also leaf-like, but they are usually bright green and greener than lichens, and they are never white underneath. They are green all over, or sometimes a little blackish, but never white beneath underneath. Foliose liverworts have small scale-like leaves and stems, so they resemble plants, but they are flat. These are foliose liverworts. And then we go to mosses. Mosses have leaves that are spirally arranged. In here, you can see one leaf and then another one that is placed a little bit farther up in the stem, but in a different position. So they are not flat, they are rounded. Okay, this is one stem, and these are the small little leaves. Both types of mosses here have the same type of arrangement of leaves, but they are different in the way they grow. Acrocarp mosses, they have upright stems that are not branched, or almost they do not have branches. They have just a few branches like in the image here. So they grow upright each stem, you can see the base that is brownish and the upper part that is usually green. While pleurocarps, they grow parallel to the substrate. They have a main stem that grows parallel to the substrate and then a lot of branches. Those branches that are lateral, they are usually, or they can grow either also parallel to the substrate or they can grow upright. So it have to be careful distinguishing these two because sometimes they can seem similar. And finally, we have also algae. Algae are a highly diverse group, as I said, and they can form either macroscopic colonies, like the one in here. They are, in general, these terrestrial algae are microscopic algae, and we will only see them if they form colonies. These colonies, like there are very typical colonies, like this one, that are surrounded by a mucilage, and then you can see them macroscopically. All the algae are embedded in this mucilage, and that's why they are macroscopically visible. Also, sometimes they form this type of crusts in the soil, blackish or brownish, that become more or less green when they get wet. So this, when you see this, it doesn't mean that the soil is dirty. It means that there are living communities in there. And we also can find them epiphytic in other organisms such as bryophytes and lichens. So very diverse group that can appear in many forms and shapes. If you want to learn more about how to identify these growth forms, especially the ones of bryophytes and lichens, you can go to this webpage, kahoot.it, and introduce this number. And you will find a tutorial with questions and different tests where you can learn how to distinguish these growth forms properly. It will be active until the 10th of March of 2021, and the link is also in the comments of this video. So I encourage you to go and try this test and see if you can identify correctly all the different growth forms, because you will need them for the citizen science program activities. Okay. So as we have seen, all the organisms in the biological soil covers are very different. We have lichens, bryophytes, algae, nothing to do with each other. However, they share some characteristics that are very important for the ecosystem functioning. These are that they are poikilohydric and reviviscent. The three of them, they share these characteristics. Let's see what does this mean. Okay, so if we think about vascular plants, they do have a cuticle in the outer layers. This cuticle is like a waterproof raincoat. So when it rains, they take a shower, but water doesn't go inside. To take water, they need roots. However, the organisms in the biological soil covers, they do not have this cover, they do not have these cuticles. So they function like sponges. When it rains, they get soaked, the water gets inside the cells. And when the air is very dry, they lose all their water. What happens when they lose all their water? They go to a dormant state. They stay inactive. If there is not enough water in the environment, they will lose their water and they will stay inactive. And when it rains, they will come back to life. That's what we mean when we say that they are reviviscent. Let's look at it with an image. Here, this is Syntrichia ruralis in a dry state, and this is how it looks, the same species, the same patch when it's wet. So they come back to life. This is what means being poikilohydric, means that they cannot control their water content, and being reviviscent means that they stay in dormant when they are dry, and then they come back to life. Why is this important? This is important because it regulates many ecosystem functions. The first one is the water balance of the soils. where they are. If they function like sponges, they can keep the soil moist for longer periods because they absorb water very fast. If you have ever watered a moss when it's dry, this process, the one I showed here, this happens in just a few seconds. They have a lot of adaptations to collect water and absorb it fast. But then they also have a lot of adaptations to lose water as slowly as possible. So they desiccate slowly. This means that they keep the soil moist for longer periods, which enhances biodiversity and ecosystem functions. But how much water? How big is this effect? If we think about this species that showed you before, Syntrichia ruralis, it's able to absorb 10 times its weight in water. It means that 2 grams of Syntrichia ruralis can absorb 20 milliliters of water. But how much is this? It's difficult to think about how much is this really. So let's think about a closer example, or let's imagine some other example. Imagine a child of 30 kilos. If it were a moss, it would absorb 300 liters of water. This means that if we put this moss child, in a pool like this, it will absorb all the water in the pool and grow big, very big. So these are some important functions, but there are many others. One of them is the protection from erosion. So this here is an image of some crusts, some biological soil covers, and if you look at it, you will see that in the parts that are brown, that are not black, there is a lot of erosion. But in the parts that are black, the erosion is minimal. So they protect the soil from the impact of the water drops. And this is important even at a global scale. Here, I will show you an example based on a study on the United States of America. So in the south of the United States, we have these soils, these deserts that are very abundant here in the south. And in the soils in this desert, you can find these communities, these black patches here. These are biological soil covers, and they are highly diverse ones. So you can find in a few centimeters a very high number of species of mosses in here and also of lichens. These diverse communities are threatened because of a number of human activities, such as off-road driving, which is very common in the United States of America, and agricultural plowing. Okay, so we have these soils that are being destroyed, and in these areas there is a lot of wind, and it is common to have wind storms like this one. So what happens with these wind storms? That they travel very far and very fast. So they can travel thousands of kilometers to the Rocky Mountains in here in the north. And the dust deposits in the snow, in the mountains, in the Rocky Mountains. What happens here is when the dust deposits, the snow changes the albedo and it melts. This produces that the snow melts before it should and changes the hydrological balance of these ecosystems, and also affects human activities because water is not flowing as it should. Okay, so we have very tiny organisms regulating large-scale processes. And actually, this is not only happening in the United States, this is a global phenomenon. More or less, we have 40% more atmospheric dust due to the desertification processes. And this same effect can be seen in the mountains around us. This is Sierra Nevada, and we can also observe this type of processes here. But there are many other ecosystem functions, such as nitrogen fixation. Biological soil covers are key to these processes. As you may know, nitrogen is essential for plants, but most of it is in the atmosphere in form of N2, which is not available for plants. Many bacteria are able to fix this nitrogen and make it available for the plants. Some plants actually have symbiotic bacteria in their roots that make this process. So they take the nitrogen from the air and they transform it so that plants can take it and use it as a nutrient. But in many ecosystems, there are not a lot of plants that can do this process. And one of these examples is the boreal forest. The boreal forest develop in cold ecosystems where the main trees are conifers. Conifers do not have symbiotic bacteria that can fix nitrogen. If we measure the amount of nitrogen that is stored in these ecosystems, we will find that the amount is about 3.3 kilos for hectare and year. However, there are just a few species of nitrogen that have nitrogen-fixing symbiotic bacteria, and these account for 0.5 kilograms of nitrogen for hectare and year. So, where do this amount come from? We're missing 2.5 kilograms of nitrogen per year in this forest. Where does this nitrogen come from? Comes from the free-living bacteria that live on this moss, Pleurotium screveri. This is one of the most common mosses in the northern hemisphere. It's widespread, and the bacteria that live as epiphytes on this moss are responsible for fixing the nitrogen that the forest needs to grow. So again, very small organisms, bacteria, mosses that drive ecosystem functions that are important globally to maintain healthy ecosystems. In fact, 40% of the biologically fixed nitrogen is fixed in the biological soil covers. So again, globally important functions made by really small organisms. The problem here is that we do not know a lot of things about these communities. We know much more about vascular plants and about many other organisms. We do not know where are they and what are the drivers that drive their diversity. And that is why we designed this citizen science project. So we called it soilSkin because biological soil cover the soil like a skin, and we want to map the distribution of the biological soil covers and their functions. We have been making a lot of measures of their functions in the laboratory, but now we need to map their distributions to be able to relate these two things and evaluate their functions and also their vulnerability to climate change. This is the research team. We are a lot of women, actually, a nice representation of people from many places. So including Portugal, Spain, and the 5 corners of the Iberian Peninsula, which is really nice. And now I will talk about the second part of the talk. We'll start, and which is about the citizen science project. What to do if you want to participate. We have developed a mobile app for smartphones that allows to record the biological soil covers and take pictures of them and fill a form with the information on the biological soil covers. And in each site, you will be required to make a transect, a linear transect, and take 3 pictures. Every 15 meters, you will have to take 1 picture. So the best way of doing it is to go to an ecosystem, to walk around, and to find the place that has the best communities, the ones that look nicer, that are more diverse or more abundant. and that will be the first recording place. You take a picture, you fill the form, I will explain later how to do that, walk 15 meters and take the next one. Again, fill the form, walk 15 meters and take the third one. Ideally, you do this in an homogeneous ecosystem. But if it's not possible, that's also okay. So let's talk about the mobile application and how it works. The mobile application is called BryoSoil, and you can find it in Google Play. Also, at the end of this presentation, you will find that I will show the web page of the project, and you can find a link to the app in there. First step, of course, is to download the app and to register. If you are correctly logged in, you will see your username here. Please remember to fill all the fields, and also remember that the fields do not accept any special characters, so no tildes, not slashes or anything. Okay, so also you have to remember to turn on your device's GPS, because the location of the pictures is essential for us to be able to map the biological soil cover distributions. Once you're logged in, if you are in a nice place, you have found amazing biological soil cover communities, you tap on this symbol here, the plus symbol, and this will lead you to the first register. This screen here, you tap in the picture, and this will lead you to your camera. And you will see that in here, down below, there is a picture of a euro. So you have to put a euro in the soil, and then you need to move up and down the camera so that you see the euro more or less in the soil, more or less the same size as the one in the image. That way, everybody takes the images from the same height. Once you have done this, you just click in here and you will take the picture. Once you have taken the first picture, you have to fill the form. To do so, you tap in here where the pencil is, and you have to fill two parts of the form. First, the type of the ecosystem. It will pop, and when you click in here in type of ecosystem, there will be a popping window that will allow you to click on several options. So choose the one that looks like your ecosystem, perhaps a forest or whatever you think your ecosystem is. And then you have to fill or estimate the cover of the different growth forms of mosses, liverworts, and lichens. So let's see how to calculate or how to estimate the cover. Imagine that you are in a community that have here in the center 50% of Pleurocarpus moss and 50% of Acrocarpus moss in here. So you will write down, or you will fill these two fields. There is a field for Pleurocarpus moss in the app and one for Acrocarpus moss. And there is a field for total mosses, which is the sum of these two. Then for lichens, we will do the same. First, estimate the amount of fruticose lichen, and then the amount of foliose lichens, of crustose lichens if there are any, and fill the fields of these two, and then sum up the two and fill the total lichens. Finally, there is a field for bare soil. In here you can see that it is very common to have some soil without any cover, so you will write down that too, and you're done. You can go to the next, walk 15 meters and take the next picture and repeat the process again and once more. In here I just show a real example where you can see more or less where are the lichens and where are the mosses and the bare soil. And our euro that is necessary to do the transect. So just a few comments on the application. It has some extra features. You can see a map with where you can explore what other people has uploaded and where do we have data points. But I would like to point to something that is very important. In here there is a field that is called connection. When you're in the field, it is you can work offline. If you tap here on connection, it will pop up a window and you can choose to work offline. I strongly recommend working offline when you are in the field, because if the connection is bad and the images start uploading and they do not get to the end, or they do not completely upload, they can get lost. Okay. It is very important then that you remember to upload the information when you get home. You can do that in here, in this part, in here in SceneMBR. So click in SceneMBR to send the information, and you will see all the images that you took and save them or send them. Also, it is very important, because many users forget about it, to save all the information. Even if you don't upload it, you have to save the information at every step. To start a new transect, because you have changed the ecosystem or moved to some other place, you just have to click here again, and the app will ask you if you want to delete the previous transect. This will only delete the transect from the screen, but if you have saved it, it will stay in SceneMBR. Although the application is in Spanish, all the information about the application is in English in the web page of the project. So you can download everything in English. The explanations, the tutorials, and also a sheet with images for all the ecosystem types and definitions for the ecosystem types, as well as for the growth forms. So if you forget the names of the growth forms, so go to the web page and explore all the resources that you can find there. So finally, I just wanted to make a summary. We have been working on this project for 1 year now, and we have been in 9 schools and worked with 10 different collectives and associations, and we have given 40 workshops where more or less 700 people have participated. Our input is really good. We have more than 300 transect, but we still have a lot of work to do if we really want to map these biological soil covers. So please help us fill the knowledge gaps, and thank you very much. If you want more information, we have a very active Twitter account, or again, just go to the web page, because there is a lot of information here. And of course, you can always write us and ask any questions about the project or doubts you may have when doing the transect. So